All right, welcome everybody. We're going to get started. It's great to have Dr. Pedro Medina with us today as our seminar speaker on HIRM. This is our second hybrid seminar, so we'll present both to an audience in the room here at Harvard Medical School, but also to all of you on Zoom. And it's great to have so many people attending. So thank you very much. So Pedro is currently a tenured full professor in the Department of Biochemistry and Molecular Biology at the University of Granada in Spain. He's also a group leader in the Center for Genomics and Oncology Research, or the GENYO, also in Granada, Spain. And he's an expert on mutational analysis in cancer, in lung cancer, and now more recently in leukemias and lymphomas as well. So Pedro received his undergraduate degree from the University of Granada, so in a way he's gone home. He then got his PhD at the Autonomous University of Madrid, where he was the first to show mutations in BRG1 and BRM1 in cancer. Of course, that's an exploding field right now. And then ended up actually doing his postdoctoral training in my lab at Yale University before settling back in Granada as an assistant professor. And now, of course, he's a full professor there. So it's great to have Pedro with us. He's going to tell us about recurrent splice-site mutations affect key lymphoma genes. So welcome, Pedro.
Well, thank you so much, Frank, for letting me speak today. I'm going to talk about our last results on recurrent splice-site mutation affecting key lymphoma genes. As Frank said, I'm working in the University of Granada and the Center for Genomics and Oncology Research. And this will be the topics of this talk. I will talk about diffuse large B-cell lymphoma, just briefly, to show the facts and the definition of this disease. Also, I will talk about the analysis of the splice-site mutation in a cohort of 1,001 patients. I will talk about the functional analysis of recurrent splice-site mutants protein. Especially, I will talk about two of them, CD79B and BCL7A. And finally, I will talk about the conclusions. Also, I will use this top panel as you can follow easily the talk. So firstly, I will talk briefly, this is a fact sheet, what is the diffuse large B-cell lymphoma. Well, this is a solid tumor of B cells that is named by the pathologies for the morphology that the cells have under microscope. It originates from the lymph nodes. Here we can see the structure of a lymph node, and it is created in the germinal centers. Although the origins are not well understood, it is the most common type of lymphoma and represents approximately 1/3 of all the cases. The annual incidence in US is around 7 to 8 cases per 100,000 people per year. And also it occurs primarily in old people, although we can find it also at other ages. Well, as other many tumors, diffuse large B-cell lymphomas have clinical changes. Actually, is the most common type of aggressive lymphoma. That means that it's grows very fast and it's need a fast treatment. The treatment is mainly, the first line treatment is R-CHOP, that is a combination of 5 drugs in which can benefit about half of the patients, but its survival, 5-year survival is lower than other lymphomas. Also, It is a heterogeneous group of disease. It are difficult to classify, and this is one of the main challenges that the clinicians have, because this heterogeneity poses a major barrier for understanding the disease and also for choosing a good therapy. So many efforts have been done to profile this disease and to get RNA and DNA profiles to study and to understand better this disease. Actually, a few of big efforts have done. This is one of them, what was published by the Duke University in 2017, and it is an amazing job that try to to characterize how is the disease and what we can do for a better classification. Yes, this is because, as many other cancers, the pathologies only have the microscopy just to classify the tumors, and for them it's difficult to figure out well, tumors that looks like similar. Well, they need tools, genetic tools, as for example, we can distinguish easily an node and O butterfly that looks alike, but if we can able to sequence them, we can distinguish them like very clearly. And this is have some challenges, and these have been improved, although there is of course room for improvement, since most of the efforts have been done studying protein coding sequences that are well, the mass study, because commercial exome sequencing are well, the most of the content are through these sequences. Of course, other sequencing, non-coding sequence might be important that having not studied for these or other major efforts, and some of them are actually not being studied, although they have information, because the commercial exome sequencing have, for example, intronic splice-site sequencing that have been discarded for many studies, and actually are important to understand the disease. Well, splice-site sequencing, this is a summary of how they look like. We know that splice sites and splicing is a very complex process, and this is just a summary, but basically is to remind that there is exome sites in the splice site. There is an exome portion and also intron portion, and many studies ignore these intron mutations when they can make a big impact on the RNA maturation. I mean, the mutation in these introns, intronic part of the splice site, can make an impact that is well, considerable. So we, in that large cohort of more than 1,000 patients, we did analysis rescuing these intronic splice-site mutations, and we found 29 genes that had recurrent somatic splice sites in at least 5 patients. Most of these splice-site mutation generates aberrant maturation, and we know that because we have the RNA sequencing, so we could analyze what is going on with these DNA mutations. And importantly, most of the genes are cancer related. So it it is really interesting, and actually for me it's really fun to figure out what is going on with these splice-site mutations, because are like really difficult to predict. Because once you have a mutation in one of these splice sites, many things can happen during RNA maturation. You can have intron rotation, cryptic splice sites, exon skipping, and overall the protein most of the time are severely damaged, or at least they can have an impact in their function. I'm going today to talk about two of these major splice-site hotspots that my lab have studied during the last years. That's where the hotspots splice-site mutation in BCL7A and in CD79B. These mutations are hotspots, as I said, and increment the mutation rate a lot. Actually, 44% of this splice-site mutation correspond to the BCL7A mutants and 32 for the CD79B. I will start talking about this last gene, CD79B. That is a gene that actually is well known if you study lymphomas, because belong to the BCR receptor. Approximately 1.6% of the diffuse B-cell large lymphomas display these mutations, and is a mutation in the splice-site donor that makes like to join together exon 4, 5, and all the intron that retains. That's why it is called this alteration, intron retention. Well, this is, as I said, this gene is very well studied in lymphomas. Actually, it is part of the BCR receptor that is composed by the heterodimer CD79B and CD79A, and AGG. It can be AGM or EGG. Is an immunoglobulin, and this receptor altogether are able to recognize the antigens and to start a signaling that helps to the B cells to do their work. As I was mentioning, this hotspot that we found, it is around here, and is disrupt the item motif. Actually, this splice-site hotspot is, well, it was described by us, but the most well-known point mutation and the other hotspot of the protein is this Y, this tyrosine in the position 196. That was like previously characterized, and it's affected the item and domain. The item domain is the cytoplasmatic domain that help to the BCR receptor to do the signaling. And then this is that one that we can see here, that is cytoplasmatic. And our mutants, as I said, it was around here, and well, the proteins are translated without the item domain, and these have some consequences that we analyze in comparison with the other point mutation that was previously characterized by the Louis Stout group, and well, we made this construct with the different mutants. This is our mutant. As I said, this green stuff will be the intron retraction. And during the translation, few codons after that, we can find here a stop codon that makes this aberrant protein and create some changes in the biology of the B cells. One of the well-known changes were that the BCR receptors are more expressed in the membrane. And we observed that it was the case for the previous mutation that is displayed in blue, the previous mutation, and our intron rotation has similar effect because both of them have unfunctional item motifs. And also, well, this it is thought that is because these mutants inhibit the degradation of the complex, and also it was observed that the or mutants also have a overreactive BCR activation that it is displayed as we observe it here by an enhance of the N-kappa-B pathway and N-kappa-B pathway that have our mutants. So, well, we could characterize this new mutant, and it has quite interesting clinical applications, especially because we enhance, we increment 32% the mutations of this gene, adding up our splice-site mutants. And actually the clinician rely for the new classification of the lymphomas on the mutation in CD79B. As you can see here, the mutation in the CD79 are important to define the MCD subclass of diffuse B-cell large lymphoma. And this clinical group is characterized by the improved prognosis, but a good response to the BTK inhibitor ibrutinib. So having these new mutants analyzed, we can help to a better classification of the tumors and also to a better treatment of the patients. And now I'm going to talk about this is a completely different gene that is nothing to do with the receptor, that is BCL7A. BCL7A, as I mentioned, is also a gene that have one of the most prominent hotspots are the splice-site mutation that could help to identify 44% of new BCL7A mutants. Actually, we didn't know much about this gene until our lab studied functionally. And around 3% of diffuse B-cell large lymphoma display mutation in this gene. And this mutation is the consequences are slightly different. When we have a mutation in this splice-site donor that is between the 1 and the 2nd exon, it activates a cryptic splice site that is inside exon 1 and makes a mutant protein that is inflamed and is aberrant, is without 27 amino acids. So we call that protein delta 27. Well, BCL7A is a member of the switch-SNF complex. As many of you know, switch-SNF complex is a chromatin remodeling complex that uses the energy of the ATP hydrolysis to modify the interaction between DNA and histone and make available or not the information to the cell and to the binding of transcription factors. So it makes a big impact in transcription. Well, switch-SNP complex, nowadays, it is well known that is important in cancer because many of sequencing studies have found recurrently mutated. Actually, I did my thesis studying BRG1 was SMART4 unit, and we found the few primary tumors somatic mutation in the lung cancer. And actually, this review that is actually from a group that have made seminal contribution of switch-SNP complexes and cancer, that is here in Boston, the group of Sigal Kadach, have recollected all the mutation during this review, but BCL7A, that is the gene that I am talking about, was not depicted there because, well, this review was done before of our publication, but maybe in the next review we can see also an arrow here saying that BCL7A is also recurrently mutated in diffuse BCL large lymphoma. We started to study BCL7A doing a study in a collection of 41 cell lines. And on this study, the cell lines are here at yellow, we can find a mutation pattern. This is a clear marker, and is like all the mutations are focusing in the exon 1 or in the initial part of the protein. So we try to look for the mutations in the patients. Yeah, but actually these mutations importantly are biallelic mutations. These profiles are the one that we found in tumor suppressors genes. And most of the cell lines have like 2 hits, one in each allele, and to make no available wild-type BCL7A for the cell. But as I was saying, we try to see if in patients we can see something similar. And actually, we have a very few patients, but we found also a similar pattern that we found with the cell lines. But of course, the patients, the number was not enough. So we tried to analyze and to see if our observations were in the measures efforts, well, that actually have access to many patients, more than we can have it. So we did our own analysis in these 2 seminal papers that study the genetics of diffuse B-cell large lymphoma, and actually we had to reanalyze their data because they have excluded the intronic splice-site mutations. But when we did this reanalysis, we found, as we observed in the survey that we did in our tumors and in our cell lines, that it was, I mean, all the mutations were concentrated in the exon 1. This is the exon 1. And the main hotspots were the intronic splice site, that these autos were ruled out because we're in a non-coding sequence. And actually, this is another way to represent the data. And here we can see the clear hotspot that we found. We found one hotspot in the initial codon that makes no protein at all. I mean, this mutation will not make a protein. Another is a point mutation. Another hotspot is a point mutation in the codon number 11. And the major hotspot, the one that we are talking today, is the splice-site mutation that by far is the most recurrent one and accounts around 21.1% of the mutation that we can have it in BCL7A. As I mentioned it, well, this is how the biology of the RNA maturation of BCL7A looks like. It have an isoform, and also when we have a mutation in this splice donor, it's activate, as I mentioned, a cryptic splice site and makes a protein that is inflamed. It is translated, but it has lost 27 amino acids, and that's why we call it delta 27. So we didn't know if this protein have an impact in the tumor phenotype. So taking the advantage that we have strong models to work with cells that have these double mutations and makes no BCL7A wild type at all, we use it for our analysis. The cell lines were OCL1 and BAL. And basically, we try to figure out how the phenotype can be revealed. And actually, it took us a while to figure out how to reveal the phenotype, because it was not an easy phenotype to reveal. But basically, how we did it, it was like making competition assays. Briefly, these computational assays, we mark our cells or the study of the cells that we would like to study in green, and it could be a wild type, a delta 27 or an empty vector, and we mix it with the not transducing cells, and with the time we'll see how they competed and if the green cells go better or no better than the non-transducer cells. And actually, we did that using, as I said, the empty vector, delta 27 and wild type. And we, in this way, we could reveal that the expression of just the wild type BCL7A makes the cells less competitive. And of course, this is an indication that BCL7A has tumor suppressor functions. And again, we did that in this another BAL cell line that displays similar phenotypes. To go further and try to see if this is happening also in vivo, we did a similar experiment using mice in which we injected in the tail BCL7A, delta BCL7A, and the empty vector. And these cells were previously prepared with luciferase, as we can easily track using EV spectrum imaging. And again, displaying similar results that we found in the competitive assay, the cell lines that express or the empty vector or the mutant are able to grow much more than the cells that are restored the BCL7 expression, displaying a tumor suppressor feature of this gene. So why these delta BCL7A were unable to develop these tumor suppressor activities? So basically, here we can see how AlphaFold represent the BCL7 protein, and this will be the wild type and the mutant. And the mutant, as you can see, it hasn't have this portion, and that is like a little hand that it have in N-terminal region. So we asked if, since BCL7A is well known to be a subunit of the switch-SNF complex, we asked if was binding to the switch, this new protein was able to bind to the switch-SNF complex. So for that, we did a simple experiment, just an AP for SMART4. That is the catalytic subunit of the switch-SNF complex. And we observed that this catalytic subunity, when we did an IP with BCL7A, of course, SMART4 was able to pull down, but with the mutant was not able to pull down. So we concluded that the mutant was not able to bind to switch-SNF complex, making the switch-SNF complex unfunctional. So to go further and analyze if the switch-SNF complex was able to scaffold without BCL7, we use another cell line that is HPL1. This line is a cell line that have homologous mutations homologous deletion of BCL7A, so there is no any mutant inside. So we did similar experiments, restoring the wild type at the mutant delta 27, and then we analyzed the proteomics using LC-MS to find that the switch-SNF complex was actually forming without BCL7A, but of course BCL7A was not binding. And this is another way to see it. Here we can see that the wild type BCL7A, when we restored the wild type 1 and we do a pull down of BCL7A, all the subunits are pulling down together with BCL7A. But when we did exactly the same with the delta 27, none of the subunits are pulling down. That means that, again, the delta 27 was not able to bind, but this experiment let us think that the switch-SNF complex was able to form, but without BCL7A. And this will be how the model works. Here are the delta 27 mutants that are able to form the switch-SNF without the delta 27 BCL7A that are not able to bind it. And actually, these results were strongly backed up when few weeks after publication of the leukemia paper, raised this another paper for the Sigal-Kadoch group, the ones that I mentioned that have made a similar contribution in the field and actually are working here in Boston. And in that group, they characterize the endogenous switch-SNF complex. This is BCL7A, close to the nucleosome and the DNA. These are the histones. And the researchers found in the terminal and that in this portion, BCL7A are binding to SMART4, to BRG1, the protein that we found. And actually, when we see what are the amino acids that were removed in our tumors, they were here, the tumors, the amino acids that were bound the switch-SNF complex. Well, and finally, we would like to figure out how or what pathways are disrupted after BCL7A mutations. So we restored BCL7A and the mutant one, the wild type and the mutant, and we compare the difference between them to figure out the pathway that were affecting. And taking the pathway that were affecting in our two models, the common one, we found that the regulation of the B cell activation were the pathway clearly affected. And this, of course, is a great news, because it could be another pathway, but we are working with B cells that are, and this pathway are frequently altered on the tumors. And when we go to figure out genes that can explain these tumor suppressor properties, we found some of them that can be or can contribute for this tumor suppressor phenotype, like P21, TP63, or TRIP2, in both cell lines were altered, and could be genes that can explain this suppressor tumoral phenotype. Right now, we continue working on the mechanism, but trying to, because the former experiment that I display could be can be done directly, but also indirectly through BCL7A. So to figure out what are exactly the direct mechanism, we are currently doing ChIP-seq and ATAC-seq to figure out what are the specific pathways that are altering in this B-cell biology activity that we found in our expression profiles. And that's all. I would like to display this take-home message. Firstly, that the splice-site mutation can contribute to the alteration of K genes in lymphoma, just as I have exemplified with CD79B and BCL7A. Also, that splice-site mutants can help us to determine the functionality of protein domains in cancer, as we have seen with BCL7A, but also with CD79B. And also, it will be important if you, I know that most of you that are in the RNA are aware that the mutation in the non-coding sequence are important, but if you are managing a project, be sure to not discard these non-exonic regions, because it can be, if you do, you can lose relevant information. And that's all. I would like to acknowledge the research of our PhD students, and actually this work has been done by mainly by two PhD students of my lab. The first one is Carlos Baliñas, that now is currently in Paris working in the Pasteur Institute. He has done most of the work that led us to publish the leukemia paper of BCL7A, and also another talented student that is going to read his thesis on September, and is Álvaro Andrades. Álvaro is the first author of the blood, and he is done an amazing work together with Carlos, that is a faculty of my department. And also I would like to acknowledge to our collaborators in the THEMA, in the GENYO and in the GENIC, and our funders, especially an association for the research in pediatric leukemia, Héroes contra la Leucemia, that is helping us to fund some projects in the pediatric side of the research line of my lab, and also to motivate us to study this kind of disease. And that's all. I will be more than happy to take your questions.
Thank you very much, Pedro. That was great. We're going to see whether there are questions in the chat, but are there any questions from the audience, from the floor? But also, if you're on the call, please feel free to unmute yourself and ask your question. But we have a question from the floor right now.
Pedro, when you said like you reanalyzed the genome and found like 6.6% of the patients who have the BCL7 mutation, like this patient had like other mutations too, or this mutation could explain like the phenotype? And the other question is like this mutation could be linked to any drug resistance to like the therapy.
Right. With the BCL7A, you mean, or with the CD7?
BCL7 mutation.
Yeah. So far, we don't know anything about the drugs that can be influencing this gene, but it's a hot topic right now. There is a lot of drugs that are developing against switch SNP components, and it could be an interesting point to do and to see if these mutant are actually one of our research lines in surveying some of these drugs that actually are a hot topic right now.
And the 6.6% patient, they have like other mutations too, or just this mutation?
Yeah, Ayman was talking just about this mutation, and they have another hotspot mutation. I mean, the BCL7A have 3 hotspot mutation. One is the codon 1, another one in the codon 11, and this splice-site mutation that is the more frequent one. And that is the one that I was talking about, that it was completely discarded during the pipeline that the other guys did, because we're just in the introns, and these guys just look for the sonic alterations and discarding intronic mutations.
So there's a question in the back there.
So you also said that this particular mutation is found across a variety of tumors. So in different tissues. So,
no, I mean, these BCL7A mutations are not observed in other solid tumors, just in B cells tumors. Actually, they were found also, I think, that in another B cells tumors like follicular lymphoma, but not in another solid tumors like lung cancer. All the tumors looks like that have a preference mutation rate in one of the subunits. Ovarian is also very highly mutated in one of the other subunits, that is RE1A. But in these lymphoma and B-cell tumors looks like are concentrated in BCL7A and also other subunits. I didn't talk about the mutation in other subunits of the switch SNP complex, but the subtype, that is the GCB subtype of lymphomas that have up to 50% of mutation in the switch SNP complex, that's huge. I mean, BCL7A is contributing with a percentage to that 50%, but overall, switch SNP complex is having a deep impact in lymphomas, and overall, it is calculated that around 25% of all the tumors in human tumors I'm talking about have mutation in one of the subunits of the switch SNP complex. So that makes one of the functional entities in the cell more frequently mutated across all human tumors.
One, do you see any other mutations that are always associated with this particular mutation? Like, because you found a lot of other mutations in these patients. So are there any particular type of mutations that occurring?
You mean concurrent mutation that happen at the same time?
Yeah, in the same patient.
Yeah, in the same patient. Yeah, it's a very, very nice question. We couldn't see any of this kind of concurrent mutation because these analyses are tricky. One of the trends that we found strongest, but without to be significant, were ACG2 mutants that are ACH2 is a polycomb complex that is related with the switch SNF functionality, and we observed that in a trend, but it was just a trend, not was significant. Otherwise, that this mutation, there is no significant concurrence.
There's a question in the chat. Can you read it there? Are paralogs BCL7, B, and C ever expressed in diffuse large B-cell lymphoma? And are they also targeted with mutations at splice sites?
This is a very, very nice question. BCL7 and B and C have not found mutated nor very expressed in diffuse B-cell large lymphoma. So this is an excellent question that we pursue, and we couldn't find any mutant, and actually were few expressed in these lymphomas.
So I guess one corollary of that is that if you could turn on those paralogs, you might be able to suppress the tumor.
Yeah, it will be a good therapeutic approach. Maybe try to push the expression of these.
And then I think the next question is, how much restoration of these would you think you would need to achieve a therapeutic effect?
Well, yeah, it is. It is difficult to figure out. It's true that, well, yeah, I don't know. Of course, in the models, they are synthetic, and we don't know how will happen in the patients. But of course, it can be a therapeutic approach that we can pursue, but we don't know.
I have a question. So you showed that in the BCL7 loss, you don't have a defect in switch-NIF complex formation. But have you been able to test activity of switch-NIF without BCL7A? For example, does it fail to shift histones on chromatin?
It's a very nice question, but yeah, I'm not expert in this biochemical analysis, and I don't know how to do it. But of course, it will be a nice way to figure out. It just was that BCL are tracking different genes or are making a different way to remodel chromatin. But of course, I'm more, my knowledge is more around the genetics, but of course the biochemical way of this non-BCL7A switch-SNF complex could be also interesting to see and to study.
And so, I mean, you showed that, for example, P21 looks like it's dramatically affected in expression when you express wild type versus the mutant BCL7A. Is that the sort of gene that you think might be being affected by switch-NIF itself? But switch-NIF is actually directly affecting the expression of P21, and that BCL7A is sort of allowing that to happen?
Yeah, it could be. That's why we are trying to find the direct effect of the binding of the switch-SNF. The problem that we have with the study of the switch-SNF is because this system that makes covalent attacks on the DNA, it is very difficult to track their whereabouts. We don't know exactly how they are binding to the DNA and are very difficult to assay. But yeah, combining the CHIP and ATAC-seq, that are assays that we can have been helpful to see changes in the chromatin structure. Maybe we can define if, for example, P21 is one of the genes that are affecting.
Have you compared EBV positive DLBCL with EBV negative DLBCL, looked at like mutational load and changes in mutational patterns?
Excuse me, what?
So is there a difference in splicing in EBV positive versus EBV negative diffuse large B-cell lymphomas?
EBV?
EBV, Epstein-Barr virus.
Ah, okay. Yeah, actually, a few days ago in the Bay Archive, I saw a paper that showed that Epstein-Barr tumors are also having a mutation in BCL7A. So I'm pretty sure that it could be most of the BCL or B cells may have mutations in BCL7A. We also observe, and my lab is working, that BCL7A is also silenced by promotherapy methylation in another B cells tumors. So I think that these alterations are also important for B cells.
Are there any other questions from the audience? Anybody online? This is your last chance. I think that's all in the chat. All right, so if there are no more questions, let's thank our speaker one more time. Oh, there's another question. Okay, sorry, one more question from Mariella. Any correlation or anti-correlation between BCL7A mutations and EZH2 activating mutations?
Yeah, actually, this I was mentioning that before, or I tried to say that. I'm sorry if it was not clear enough. We found a trend between these BCL7 mutations and EZH2 activation mutation. It was just a trend. It was almost significant. I think that was 0.07 or so. It was close to the significant. But yeah, we think that there is maybe a correlation, and it's interesting because there is some drugs that are currently using that are inhibitors of EZH2 that can be maybe benefiting the BCL7A mutants. We are studying that in the lab, but currently we don't have any results. But yeah, looks like at least a trend.
All right, well, that sounds very exciting. Well, thank you very much. Thanks everyone for your attendance. And just a reminder that this is our last seminar for this academic year. We'll start again in September of 2022. So we're taking a few months off for the summer. I hope everyone has a very productive summer. So thanks for all your support for this past year. And thanks to Pedro for coming away from Spain. It's great to have him here. Thanks, everybody. See you later.